Hello, and welcome to our lecture on muscle tissue. Let's go ahead and get started. So hopefully this should be familiar to you. Over here on the left-hand side, we have skeletal muscle. In the middle, we have smooth muscle. And last but not least, over here on the right of screen, we have cardiac muscle. So we still need to remember the basic properties. There's three types. Remember that skeletal muscle and cardiac muscle are both striated because we can see those striations here and here. In this lecture, we're going to figure out where this actually comes from and why actually smooth muscle is non-striated, which is interesting because the basic components that make skeletal muscle and cardiac muscle striated are also found in smooth muscle, but they have a slightly different arrangement, and that is what difference in arrangement is what leads to no striations being visible. We have squash nuclei in the periphery for skeletal muscle. Cardiac muscle, we have more centrally located rounder nuclei. Smooth muscle, we have these fusiform shapes, right? So they're spindle shaped, they're pointing to either end, also centrally located nucleus. Cardiac muscle, smooth muscle is involuntary, while skeletal muscle is voluntary. So we still need to remember all those little bits of data. And in the lab test for this section, you may come across slides of this tissue type, and we still need to remember all that information from our previous section. Okay, so as I just went over, three basic types of muscle tissue: skeletal muscle, cardiac muscle, smooth muscle. There are more than 700 different skeletal muscles that make up our muscular system, which is, important for body movement. Right? There are other characteristics we do need to know about muscle tissue. So first, it is excitable. So it has a characteristic of excitability. So depending on what type of muscle you have, when it receives a stimulus, it gets short. Right, it's going to contract. Now, in the case of a skeletal muscle fiber, we usually have a direct neuronal connection. Right, so a nerve is going to zap the muscle muscle is then going to contract. In the case of skeletal muscle and cardiac muscle, we're going to see that there's an exchange of electrolytes, there's an exchange of ions from one cell to a neighboring cell, and that is going to signal for the contraction of that second cell. More on that later on. Now, contractilicity, so these muscles, what do? They get short, right? I already mentioned that. So when they are stimulated, you're going to see the overall length of the cell shorten, and we'll take a look at exactly how that happens in a little bit. Muscle is also elastic. So when the signal to contract is removed, the muscle will relax. It will go back to its original length as soon as that stimulus to produce tension has been removed. The last one is extensibility, meaning that you can stretch a muscle cell a little bit, and thus an entire muscle, a little bit longer than its resting length, right? So it can be stretched beyond its normal resting length. Okay. So we should remember, just like bones, each individual bone is considered an organ unto itself. The same is actually true of skeletal muscles, like your biceps brachii. You're going to find all four basic tissue types. Skeletal muscle is striated, and in most cases, skeletal muscle is attached to bone. Both ends of the muscle are attached to different bones. Now, there is an exception to this, like the muscles in our face. They will usually attach to bone on one end, but they will attach to your skin or the integument on the other end. That's how we're able to make all of these weird facial expressions, because we have skeletal muscle yanking on the skin of our face, while the other end is attached to bone. All right. Functions of skeletal muscle. Obviously, this is very important for body movement, how we walk and how we talk. Maintenance of posture, right? Being able to keep our backs straight, keep our hands off the floor, right? So basically, to be able to stand up upright, that takes quite a lot of muscle tension. We have temperature regulation. You not normally think of this, right? There are actually two factors for this. One is simply we have more mass covering or being a part of our body covering our visceral organs that provides some amount of insulation. But also if it gets really cold, what happens? Your teeth start chattering, you start shivering, right? That's your skeletal muscle having repeated contractions as a way to generate heat. Storage and movement of materials. So storage, what are some of the things you store in muscle? Glycogen is actually stored, which is an energy reserve, can be stored in muscle. Movement of materials. So later on in the circulatory system, we're going to learn that the action of skeletal muscle can actually help return blood to your heart. It's a way to, if you're inactive, if you're sitting for long periods of time, perhaps you notice you've been on an airplane, your feet are going to swell up. Right? What do they say? I have poor circulation. I need to go walk it off. Or I need to go walk around, because the action of your skeletal muscles will help pump blood in your veins back to your heart. But we'll get into the details of that later. And lastly, support, right? Just structural support for the body. One of the main reasons we have abdominal muscles is to simply prevent our visceral organs from spilling out all over the place, right? We don't have bone there to keep that stuff inside of us. That's one of the functions of skeletal muscle. Okay. Let's start talking about the parts of skeletal muscle. Each muscle is comprised of muscle fibers. Remember, a skeletal muscle cell is sometimes called a muscle fiber because it looks like a fiber. It's really thin and really, really long. Now, these fibers, these skeletal muscle cells, are bundled together in structures called fascicles. So a fascicle is a bundle of muscle fibers, which is simply a group of skeletal muscle cells. So, excuse me. Inside each fiber, you will find structures called myofibrils. Whenever you see this prefix myo, it means having to do with muscle, right? So myofibrils, these are going to be little structures that are inside skeletal muscle cells. Each myofibril is made up of myofilaments. So myofilaments are smaller, they're a part of myofibrils. So in our order of structure here, we have myofilaments. The thinnest of these structures is actually made of protein. So myofilaments group together to make myofibrils. Myofibrils are found inside a skeletal muscle fiber. Lots of skeletal muscle fibers packed together are called fascicles. I should point out, any of you who ever enjoyed a steak or anything like that, or cooked your own food, people talk about the grain of meat. There's like these little tiny stripes through the meat, just like you have grain in wood. Well, the grain of meat are the fascicles. That's actually what you're seeing. So when you look at those little lines or that texture in meat, notice all those lines are going in one direction. Those are the fascicles of a muscle made up of individual skeletal muscle cells. Okay, so down here at the highest level of magnification, we have one single skeletal muscle fiber. That is the plasma membrane, which we're going to learn. It has a fancy name, the skeletal muscle. But right now, this is the plasma membrane of a skeletal muscle cell. It is packed full of myofibrils. Each of these little rod-like things is a myofibril. Right? This is the peripheral nuclei of a skeletal muscle cell. Remember, it's all packed full of so many proteins, the nuclei gets pushed off to the side. So this is one skeletal muscle cell. It is packed full of myofibrils. If you were to zoom in on the myofibrils, you would see myofilaments. Okay, so we have one skeletal muscle cell filled with myofibrils. Here, each one of these little disc-like structures here is the end of one skeletal muscle cell. So what this is showing you is a whole bunch of muscle fibers all packed together. That means it's a fascicle. So over here, this image right here is the same as this image right here. So each of these circles is now one single skeletal muscle cell all packed together in a structure called a fascicle. So here, these little rod-like structures myofibrils. On this level of understanding, we have skeletal muscle cells packed together in megafascicle. Here we can see that an entire muscle is broken up into different fascicles, right? This is what's giving you your grain appearance in meat, all the different fascicles packed together. Now, we're going to go over some connective tissue that we're going to find in between cells, in between fascicles, and surrounding the entire muscle. This connective tissue is going to provide strength and support, places of attachment for nerves and blood vessels, and eventually, at the extreme ends of these two muscles, all this connective tissue from all these layers that we're about ready to talk about all join together to make a tendon, which is the connective tissue that attaches muscle to bone. Okay, 3 layers of connective tissue. We have endomysium, meaning on the inside. Mesium, again, this is sort of another root or having to do with muscle. So endomysium, this is the innermost layer of connective tissue, and it's going to surround each skeletal muscle fiber. So you're going to get a layer of endomysium, which is made of areolar connective tissue. So around each skeletal muscle cell, there's going to be a bundle of areolar connective tissue separating it from all the other skeletal muscle fibers. So let's read the little title here. Innermost layer that surrounds and electrically insulates each skeletal muscle fiber. Think about that for a second. Why would you want to insulate one muscle fiber as opposed to another one? Well, the exact hows and whys we'll get into eventually. You probably won't fully understand until you get to physio. But the basic underlying concept here is if you activate one single skeletal muscle cell, you want that one only to contract all by itself. If it was not insulated, you would end up with a chain reaction. You would trigger one skeletal muscle cell, but then if it was not insulated, it would activate all the cells around it, all the cells around that one, all the cells around that one, and then you would have an uncontrolled muscle contraction. We don't want that to happen. So the endomysium is made of areolar connective tissue, and it isolates and insulates one single skeletal muscle cell. Okay, next layer up, we have perimysium, meaning around. What is it around? This is a layer of dense irregular connective tissue surrounding an individual fascicle. So perimysium, dense irregular connective tissue, separates one fascicle from another. And then finally, the largest level, right, so going out the furthest, we have epimysium, right? This is surrounding the entire muscle. So this is a layer of dense irregular connective tissue that separates one muscle from another muscle. Sort of subdivisions of this, we have deep fascia and superficial fascia. Deep fascia is what's going to separate one muscle from another, while superficial fascia is what's going to separate muscle from skin, or the integument. Or more appropriately, it's what's going to separate muscle from the hypodermis. Okay, so if let me go back to this image right here. So we have one skeletal muscle cell down here, one skeletal muscle cell. This is the plasma membrane of that cell, which we're going to find out has a fancy name. We'll get to that in a second. Each skeletal muscle cell is surrounded by a layer of areolar connective tissue to insulate it, and that's called the endomysium. Here we have 1, 2, 3, we have many individual skeletal muscle cells. Here, the connective tissue separating one skeletal muscle cell from another skeletal muscle cell, endomysium, right? A bunch of cells together makes a fascicle. So the connective tissue surrounding an individual fascicle, perimysium. So you take the blow up here, all these are individual muscle cells. So in between is endomysium, surrounding the entire fascicle is perimysium. Bunch of fascicles put together and you get a muscle. What surrounds the entire muscle? Epimysium. So the layers of the perimysium and endomysium all join together at the tail end of the muscle to become the tendon, which is what's going to attach the muscle to bone. So here we have a whole list of structures going from the very largest to the smallest that we've discussed so far. So we can see the level of a muscle. A muscle is broken up in sections called fascicles. Fascicles are broken up into smaller components called fibers. Fibers can be subdivided. Look inside a fiber and you see myofibrils. This image is showing you the structure of one myofibril. And inside the myofibril, we can see myofilaments. And you take a zoom into the myofilaments and you find that we actually have two different kinds of myofilaments. We have thin filaments and thick filaments, so named because one is thicker than the other. We'll talk more about these in part 2. Let's continue on and discuss muscle attachments. So the end of each muscle, all that connective tissues are going to join together to form a bone, to form a tendon, and the tendon will attach to bone, or it may insert into the skin, or potentially even another muscle. Now, there we have a little word here we need to define, an aponeurosis. That's kind of a scary, sciencey sounding word, but all an aponeurosis is a flattened tendon, right? Instead of being a little cord, like a pencil, like a, like string, it's just flattened, and that's called an aponeurosis. Now, most muscles, most skeletal muscles are going to extend over an articulation. They're going to extend over a joint, right? So you're going to have one end is going to attach to one bone and the other end is attached to a different bone, which sort of makes sense because this is what's going to power the joint. This is what's going to allow the joint to move. If you have a muscle that's going to be attached to the same bone, different ends of the same bone, when that muscle gets short, nothing happens, right? Because the bone is this rigid object and the muscle's sort of pulling on this rigid object, nothing's going to happen, right? So if this is your bone, you have a muscle on either end, and that muscle's trying to connect A to B, right? Nothing's going to happen unless you break the bone, right? So we obviously don't want to do that. So normally you're going to have two bones with an articulation, and you're going to have a muscle, right, connecting to one bone to another bone, crossing an attachment. When that muscle gets short from A to B, you move those 2 points close together, one bone will move, the other bone will not, right? So this is the bone that moves, and in this example, this bone's moving, this bone's not, right? The bone that moves is called the insertion, right? So that's the end of the muscle that attaches to a bone that's moving. The bone that does not move is called the origin, or more appropriately, the end of the muscle attaching to the bone that's not moving is called the origin. So to put a little bit more clearly, the less movable point of attachment is the origin, what does not move. The more movable point of attachment is called the insertion. So we can now describe many of the characteristics of a muscle. We can learn its name. We can learn the bone that it attaches to that doesn't move, that's the origin. The bone that it does attach to, which does move as part of the action of the muscle, is called the insertion. So we have name, origin, insertion, and the final thing is the action, which is a description of the kind of motion. So abduction or adduction, flexion or extension, right? These are all descriptions of motions that are driven by muscles. Okay, so here in our sort of easy example, we have the biceps brachii. So the biceps brachii literally means bi. What is cephalic, right? Is your head. Biceps means two head, right? Brachii means of your brachium, right? So it's literally the two-headed muscle of your brachial region. So there it is, 1, 2, the two-headed muscle of your brachial region. Now, we have the biceps brachii. It's going to originate on humerus and your scapula, and it's going to insert on the radius, right? And then when this muscle gets short, this point and that point are going to move closer to each other, so that means your antebrachium, led by your radius, is going to decrease the angle between those two bones. So this would be 0 and that would be 180. So decreasing the angle between these two bones is defined as flexion. So the action, the biceps brachii gets short, brings the radius closer to the humerus. So the action, you would say, is flexion of the radius, or flexion of the antebrachium, obviously, because the ulna is going to come with it. If your radius flexes and your ulna doesn't, that's a bad day. So how would we characterize this muscle? So based on this action, flexion of the antebrachium, we know that the radius must be the insertion because it's the bone moving. And let's just call it the scapula is the origin because it is not moving. Now understand that these definitions are dependent on the actual motion. Meaning, in most cases, if I'm just standing here like this, and I make the biceps brachii short, what's going to happen? The radius and ulna are going to move, right? But what happens, for the sake of argument, that I take a giant nail and nail my hand to like a big tree. That tree is not going anywhere. And I take a cattle prod and I zap my biceps brachii and cause it to contract, right? My hand and arm are not moving, but if my biceps brachii is strong enough, when that gets activated, it's going to bring me closer to my hand. In that case, in this ridiculous example that I've come up with, radius and ulna is not moving, but the humerus is. And in that case, origin and insertion are going to switch. In that motion that I've described, the radius is now the origin and the scapula is now the insertion. Right? So that is a different situation. Perhaps something that's a little bit more reasonable is think of most people are familiar with your abdominis rectus, right? So it's a muscle that's running up and down superior inferiorly in your abdominal region. It's going to have its attachment point at your xiphoid process. Everyone should know where that is now. And it has another attachment point at your pubic bones. So when you're doing a crunch, right, you're doing like a sit-up. What's the origin and what's the insertion? Well, think about it. If I'm laying on the ground, I do a crunch, I'm bringing my xiphoid process towards my pubic bone. Okay, so that means the xiphoid process is going to be the insertion and the pubic bone is going to be the origin. Now, if you did a different exercise, right, the thing where you like grab the overhead bars, you lift yourself up, and then you raise your knees and try to get your knees to touch your chin. So now you're suspended from bars, right? You're just hanging in the air and you bring your knees up to touch your chin, right? One of the muscles involved in that is going to be the abdominis rectus muscle, but now your pubic bone is the one that's moving and your xiphoid process is not. So in that case, it's reversed. The xiphoid process becomes the origin and the pubic bone becomes the insertion. So you need to understand the motion in order to be able to determine the origin and insertion. Or you can look at the bones and figure out which one is likely to stay still and which one is likely to move. And then you can figure out origin, insertion. And from there, you can figure out the action. So you need one or the other to figure them out. If I don't give you the origin, insertion, right, and you can't figure it out by looking at it, I would have to give you the action. But if I give you the action, you should be able to figure out the origin, insertion. Likewise, if it's clear, or if I tell you what the origin, insertion is, you should be able to figure out the action. So what am I rambling on about? 700 muscles in the human body. Each one, name, origin, insertion, action. If you want to get fancy, innervation. Right? Now you have more than 700 times at least 5. Right? So now you're at 3,500. A lot of those muscles are going to have multiple actions. So now you're closer to like, I don't know, 5,000 or 6,000 bits of information. That's a lot to memorize. That's an awful lot, especially in an introductory class. So what my aim here is to give you guys opportunities to develop the skills to be able to figure out origin, insertion, and action. Now, name and innervation, you would just have to straight memorize. In this class, I do not make you memorize any innervations. You don't need to figure out, remember which nerve actually activates your biceps brachii, which nerve activates your triceps brachii. We're not going to worry about that in an introductory class. But origin, insertion, and action, I'm going to try to give you guys the skills to be able to figure them out, because then you have less memorizing you have to do. And then you can, when called upon, you can simply look at it and try and figure out what's going on. I'll give you lots of opportunities for that in lab. But to recap, what's the important thing? The origin is the end of the muscle that attaches to a bone that does not move or moves the least. The insertion is the end of the muscle that attaches to the bone that is doing the primary movement. Okay, now let's take a look at some specialized structures in skeletal muscle fibers. So as I mentioned, we have some differences here, and these different structures inside skeletal muscle cells get fancy names. I mentioned earlier that the plasma membrane on skeletal muscle cells gets a fancy name, and they're called sarcolemma. So when you see the sarco, this has to do with a structure called a sarcomere, which also is, let's just say, remember that it's related to muscles. So when you see the sarco, we know we have something to do with muscles. So the sarcolemma, lemma, like lamella, like to laminate, means a layer. Right, so the sarcolemma is a layer surrounding the skeletal muscle cell. So that's the plasma membrane in the skeletal muscle cell. Sarcoplasm, this is just a fancy name for the cytoplasm. Sarcoplasmic reticulum, right, this is just a specialized version of smooth muscle, right? Endoplasmic reticulum, sarcoplasmic reticulum. It's made specifically of smooth ER in the skeletal muscle cell. Now, these structures come together to form some other unique structures. We have what's called a T-tube or a transverse tubule. A T-tubule is a hole, right? It is a tube that is an extension of the sarcolemma. So think of the skeletal muscle cell covered in its sarcolemma, and then you get an invagination, an infolding that turns into a tube that buries deep down into the skeletal muscle cell. Those are T-tubules. So deep invaginations of the sarcolemma that extend into the sarcoplasm. Right? Form a network of tubules that enable muscle impulses to spread quickly and internally. So what exactly is going on there with muscle impulses? The details of that will wait for physio. What I do want you to understand at this point is that you're going to have a signal. What that is, we'll study in physio, but there's going to be a signal running across the sarcolemma, the plasma membrane of the skeletal muscle cell, and that signal will travel down the T-tubules and be spread out to the entire internal volume of the skeletal muscle cell. So it's a means of signal transduction. Sarcolemma invaginating to form transverse tubules, which then spread out throughout the skeletal muscle cell. Now, another structure we're going to find is called terminal cisternae. Remember cisternae or chambers? We had cisternae in the Golgi, and we had cisternae in the rough and smooth ER. Terminal cisternae, surprise, surprise, are made of the sarcoplasmic reticulum, which, as I just told you, is specialized smooth ER in skeletal muscle cells. Now, what these do is store calcium ions. The details of that, I'm going to leave to physio, right? I have to draw a line somewhere between anatomy and physio, and this is where we meet that line. Terminal cisternae are blind sacs of the sarcoplasmic reticulum, smooth ER. Often you will find two terminal cisternae next to one transverse tubule. Put these things together and you get what's called a triad, two terminal cisternae and a T-tubule. So what you're going to see is a T-tubule is a way to get a signal from the outside of the skeletal muscle cell to the inside of the skeletal muscle cell, which is right next to the terminal cisternae, which holds calcium, which we're going to learn has something to do with initiating the contraction of a muscle. As far as the functional understanding, that's it for these structures. The details of this you will learn in phys. You start learning about ion pumps and gated potentials and all that kind of stuff. Wait for that until next semester. Okay, let's go over a picture. Hopefully, should make this a little bit more clear. So this is one skeletal muscle cell. We have pulled back the endomysium. The areolar connective tissue making the endomysium is gone. So now our first layer here is the sarcolamella. An invagination of the sarcolamella makes these green guys here. These are the transverse tubules. Notice the transverse tubules are going to spread out throughout the entire skeletal muscle cell. On either side of the transverse tubule, we have blind sacs of the sarcoplasmic reticulum, right? The terminal cisternae, 1 and 2. So 1 T2 plus 2 terminal cisternae equals a triad. Tri meaning 3. 1, 2, 3. So what are the terminal cisternae full of? They're filled of calcium ions, which we're going to find out in part 2. It's the trigger to actually cause the contraction of the basic functional unit of the skeletal muscle cell called a sarcomere, which is going to ultimately lead to the contraction of the entire muscle. But I'm getting ahead of myself. Let's review structures. Plasma membrane has a special name called the sarcolamella. Infoldings of the sarcolamella form transverse tubules. Transverse tubules radiate out throughout the inside of the entire skeletal muscle cell. On either side of the transverse tubule, we have terminal cisternae, which are modified forms of smooth ER, which store calcium ions. Done. Also in this figure, we can see the myofibrils. Look inside the myofibrils, and you will see the myofilaments, thick and thin. Also, you notice there's a lot of mitochondria, right? Because muscle uses a lot of energy, you're going to need a lot of ATP. And we have a multinucleate cell, it's skeletal muscle cell. The nuclei are pushed off to the periphery. Okay. Now, getting back to this topic of myofibrils and myofilaments. So myofibrils are large cylindrical structures that run most of the length of a skeletal muscle cell, right? Run the entire length of the skeletal muscle cell. And these things can get short. When your myofibrils contract, that's going to cause the muscle fiber itself to contract. When lots of muscle fibers are contracting, fascicle contract, the whole muscle will contract. Now, myofilaments are the small protein fibers inside the myofibril. Myofilaments do not run the entire length of the skeletal muscle cell, but they're going to be organized in distinct groups or distinct regions called sarcomeres, which we'll look at in a second. As I mentioned, we have two types of myofilaments, thick filaments and thin filaments. So here is a picture of the thick filaments. Thick filaments are made of a protein called myosin. Myosin, like muscle, myo, right? Remember that root. So myosin, and we can see that the myosin proteins have two distinct regions. They have a head and then they have a tail. And two myosin molecules are interwoven together. So we have two heads and the two tails are interwoven. And then a bunch of these get interwoven into make the thick filaments. So myosin is the protein. Many myosin molecules bound together makes a thick filament. The reason the heads are important because this is where the myosin protein will bind ATP. This will lead to a conformational change, and then it leads to movement. But we'll get to that in a second. Now, the second protein we have here are thin filaments. And the protein of the thin filament is actin, right? So actin is the basic subunit of the thin filament. And we have two other proteins attached to their thin filament here. We have this green rope-looking thing, which is something called tropomyosin. Tropomyosin. And we have this little stack of pancakes here. This is called troponin. So the function of these I'll get to in a second. But basically, when actin and myosin get together, the muscle is going to get short. Remember I said you have these heads of the myosin molecule, which can bind ATP. Well, if these guys get ATP, and if they're allowed to touch the thin filament, the muscle will contract. These filaments are going to slide over each other because the heads of the myosin are going to pull on the thin filaments, and it leads to shortening of the myofibril. The job of troponin and tropomyosin is to regulate or to control whether or not actin and myosin are touching. Do the thin and thick filaments get to touch? If they do touch, movement's going to happen. If they don't touch, the muscle stays relaxed. And what we're going to see is this arranged pattern of thin, thick, thin, thick, thin. So the details of the myofilaments, thick filaments, are the larger of the two, right? They're about 11 nanometers in diameter, about twice the width of a thin filament. They're made of a protein called myosin. The myosin molecule has a head and a tail. The head is capable of cross-bridging, meaning it can bind to the thin filaments, right? When they cross-bridge, when they attach, then they can start to slide past each other, and then you're actually going to get contraction of the myofibril. Myofilaments, thin filament, about half the diameter, 6 or so nanometers. We actually have two different kinds of actin. We have F-actin and G-actin. F-actin means filamentous, like a string. G means globular. For our purposes, don't worry about the specifics of that. Just two slightly different versions of actin protein. Right? But these two guys are going to be twisted around each other. In addition, we have the regulatory proteins tropomyosin and troponin. So let's discuss what these two proteins do. Tropomyosin is going to prevent actin and myosin from touching. So the thing to remember, that the default position is on. Right? If actin and myosin touch, boom, the muscle contracts. Right? I'm sitting here fairly relaxed. Most of the muscles in my body are not contracting. So there has to be something preventing actin and myosin from touching, because as soon as they touch, boom, they're going to get short. Right? We're not having uncontrolled muscle contractions every second of every day. It's because tropomyosin is going to inhibit, tropomyosin is going to prevent actin and myosin from touching. So it's going to get in between the two and say, you two keep away from each other. So the job of tropomyosin is to prevent the interaction of actin and myosin. Okay, so if these two are always kept apart, how can we possibly activate the muscle? Well, you need to inhibit the inhibitor. So troponin inhibits tropomyosin. So remember, tropomyosin is the protein keeping the actin and myosin apart. Well, if you pull that away, now actin and myosin can touch and you get muscle contraction. So troponin stops tropomyosin from stopping actin and myosin from touching. Right? You inhibit the inhibitor. That may take a little bit of time to kind of process. You might need to read the book, read an article about it, talk to me in the Zoom session. Troponin blocks the action of tropomyosin. You take tropomyosin out of the picture, actin and myosin can touch. As soon as they touch, the muscle gets short. Okay. So one more time, we can take a look at this figure. We have myosin as the thick filaments. We have actin as the thin filaments. We have our regulatory proteins, troponin and tropomyosin. So you can see here, the way this works is the green cord, the tropomyosin is going to prevent the heads of the myosin from touching the actin, touching the active sites of the actin. Notice, look at the little guy there, calcium binding site. So the way this works is calcium released from the terminal cisternae are going to bind troponin. That causes troponin to change shape. Troponin changing shape causes tropomyosin to change shape. When tropomyosin changes shape, that exposes the active sites of actin, so myosin can bind, and then the muscle get short. Now, how did calcium get in here? Right, that's a story I'm more or less going to leave until we get to a little bit later on in this chapter. And the excruciating detail I will leave to physio. But because we've gotten this far, I'm just going to give you kind of the whole story. An action potential is going to travel down a motor neuron. At the end of the motor neuron, you're going to release neurotransmitters. The neurotransmitters are going to pass through the synapse, a gap. By the way, this is all stuff I'll talk about in a second. The neurotransmitters are going to hit the other side of the synapse, which is the sarcolemma. There it will generate another action potential, which travels along the sarcolemma, down the transverse tubules, then activates the terminal cisternae. Terminal cisternae release calcium. Calcium binds troponin. Troponin has conformational change, which changes tropomyosin. Tropomyosin moves out of the way, actin and myosin bind, and the muscle gets short. So, just for completeness, I threw that in there. I don't expect you to understand this whole story yet. Right? To get all the gory details, you'll wait to physio. But I'm just giving you a little bit of foreshadowing so you understand when we look at these structural components, where are we going with it? First, we're learning the parts. I'm going to give you sort of a generalized big picture of how they work. When you get into physio, you'll get into the nuts and bolts of exactly how this works. But it doesn't make a whole lot of sense for me to just give you a tiny little part of the story. The details of the structure are what I'm going to test you on, but conceptually, you need to understand the big picture. Okay, this is a very nice table, which is going to cover all the structures that we've covered up to this point: sarcolemma, sarcoplasm, sarcoplasmic reticulum, terminal cisternae, transverse tubules, right? Description and function, you should know all this information. Same here with the thick and thin filaments. What are they made of? What are the regulatory proteins? Nebulin, don't worry about because that's a little bit vague. Connectin, another name for connectin, we haven't talked about this. I'll show you in a subsequent slide. But another name for connectin is called titin. All right, titan means huge. It's titanic. This is actually the largest known protein. It is an enormous protein, right? And it helps basically keep thick and thin filaments in their proper orientation. I'll give you more details on that in part 2. So that brings us to the end of our first section in muscle tissue. We will continue talking about how the thick and thin filaments are arranged in a structure called the sarcomere next time. Till then, take care.